Sorry for the delay. Because of some technical problem, there is a delay. Today, a small topic is left in this unit. Plasma membrane. We'll go through it. Okay. So, plasma membrane is our fourth unit in biology. There we studied about diffusion and osmosis between the activities. So now, first of all, we should know what is diffusion. Diffusion, as already I said you, moment of concentration become equal. So that was explained by Thomas Graham. He was Scottish, both the physicist and the chemist. Thomas Graham is both the physicist and the chemist. He explained about the diffusion of different gases, and he said more soluble liquids diffuses faster than the less soluble ones. Such a law is known as, and he said that law as Graham's law of diffusion. He studied the rate of diffusion of different gases. He studied the rate of diffusion of different gases. The Scottish scientist Thomas Graham, he was both the physicist and chemist. Means he studied both physics and chemistry. He said that different gases diffuses at different rates. Rate of diffusion of gases is different in different solutions. So here, and he explained more soluble substances diffuses faster than the less soluble substances. Okay, and he proved it as Graham's. He said it as Graham's law of diffusion. What is more soluble substances? As we already studied solubility. So dissolving any solvent and solute. Dissolving the solute in the solvent, it become a solution. So more solvent if you are adding and if you are making a solution, this it will dissolve and dissolve and dissolve and it becomes saturated. So that is more soluble. So more soluble substances dissolve faster than the less soluble one. That was explained by him, and he called it as the law of diffusion. Okay, that is about diffusion process. It was explained by Thomas Graham. Then next we'll come to the next topic, that is reverse osmosis. Reverse osmosis on the other day also I explained you. It's very useful, and everybody in today's life, everybody is using this reverse osmosis process at home. So what is that reverse osmosis? Reverse osmosis is nothing but it is equal to diffusion process. Reverse osmosis is nothing but it is equal to diffusion process, but here we are using the semi-permeable membrane. Without semi-permeable membrane, this process will not take place. And this reverse osmosis, especially we are using for desalination process. Desalination process is nothing but removing excess amount of salt from the sea water, and that salt we are using for our daily life, that is for eating purpose, and the remaining water which is desalinated is become fresh and we are using for our daily life, fresh water we got. So very useful process, desalination process. Once upon a time, near the, now also they are doing near the oceans and all, what they do, just they hang up the 3-layered semi-permeable membrane, and the highly concentrated salt water, this sea water will move on to this semi-permeable membrane, and the entire salt crystals will remain here. The water will filter and come to this side. This water is nothing but it is fresh water, and the salt is collected here. This salt we use for our daily life. Okay, so that process is done, which is called as reverse osmosis process. But nowadays we are doing that process by the machine called as reverse osmometer. Reverse osmometer is nothing but the machine which is used to do the reverse osmosis process to remove the excess amount of salt from water. Okay, they are using the machines called as reverse osmometer. At home also we are using that, we call it as RO. RO, what is RO? That is reverse osmosis. In our school also we have the reverse osmosis, that is water plant we call it. Water plant, once upon a time we used to call it as purifiers. Water filters we used to call, where we used to have candles in the water filters. We used to have candles in the water filters. That candles used to absorb the excess amount of salt from the water, and it used to pass the fresh water into the next chamber, next utensil. So in such a way it is used to purify the water. But afterwards purifiers came. We call it as purite. In the charcoal machine is there. It is absorbing the excess amount of salt and releasing the water which is fresh water. excess amount of salt is removed, where we cannot drink that excess amount of salt. Even if we drink the excess amount of salt water, it is maintaining the ionic, it is causing the ionic imbalance in our body and leading to blood pressure, high blood pressure. Okay, so in such a way it is very harmful. Hence, we have to remove the excess amount of salt. How much salt is required for our body, that much only we are taking. So how we are doing that process? By reverse osmometer. Then what is the reverse osmometer will do is, it is having the machine like this, where it is having the semi-permeable membrane. Semi-permeable membrane is there in this, and from one side it is applying the pressure. It is applying the pressure, and then we are passing the water here. Water flow is here. Water flow is here, and all the impurities are removed onto this, and the water will come in this, the upward direction. Such a process is called as reverse osmosis process, where excess amount of salt is removed from the water by the machine called as reverse osmometer. Okay, so in such a way, in daily life we are using this. Every home is having this reverse osmometer, where it is doing the reverse osmosis process and purifying our water. And the excess salt along with water, not in the form of crystals. Here in oceans we will find it in the form of crystals because of evaporation process. But here at home, we cannot see any salt crystals. Salt crystals are available and salt water is available at home. We will not buy from the market. It is not happening here. That also the salt which we get by reverse osmosis process means after purifying the water, the excess amount of salt is not in the form of salt. Again, it is in a diluted form with water. It will go from another side. Such a process is called as reverse osmosis, which is very useful in our daily life. The next process is that we are going to study in our 10th class. Just this is given here because it is related to our topic. That is osmosis and filtration topic. Already we completed what is osmosis and what is filtration. How it is taking place in our physiological process. To understand that, it is given here dialysis. First of all, we should know what is dialysis. Dia means through, lysis means splitting. This is a Greek word. This is a Greek word. Dia means through and lysis means splitting or breaking down. Lysis means splitting or breaking down. Dia means passing through. So, removing the impurities by passing over to the filter. What is that filter in our body? In our filter is kidney. Kidney, this is a structural and functional unit which are called as nephrons. These nephrons are the filters which are doing the process of dialysis. But when the person is undergoing ESRD, that is end stage renal disease, when both the kidneys fail to function, not one kidney, two kidneys are there. When both the kidney fails to function, such a condition of a person is called as ESRD, or it is also called as uremia. It is also called as uremia. Okay, when excess amount of uric acid is absorbed in the blood. When excess amount of urea or uric acid is absorbed in the blood, such a condition is called as ESRD, or also called as end stage renal disease, also called as uremia condition, also called as kidney failure, During such a process, the person have to undergo the artificial kidney process. Such a process is called as dialysis. Okay, dialysis is through, lysis means splitting off. So passing the blood, when the kidneys are not functioning in the person body, outside the blood is exposed, the person blood is passed on to the machine, which is called as artificial kidney. What is an artificial kidney? It is nothing but it is dialyzer. This is a machine which is used to purify the blood, to remove the impurities from the blood. So first of all, who was the scientist who prepared this machine? The machine was first prepared by William Koch. He was the first scientist to prepare this machine in the year 1947. William Koch was the first scientist in the year 1947. He prepared the dialysis machine. And this dialysis machine is working under two principles. What are that is? Osmosis and filtration. Osmosis and filtration. These are the two processes which we studied already. Osmosis and filtration. These are the two processes, osmosis and filtration. reaction is taking place in this dialysis. In our natural process, physiological process, also these two processes are taking place. So, how this will happen? How this will happen? This will happen over the machine called as an analyzer. The machine is there where the person blood is passed with the help of artery. Person blood is infused onto this machine, removed from this person body with the help of artery, and it is passed over the dialyzer machine. This dialyzer machine is having the semi-permeable membrane made up of cellophane. Cellophane. This is cellophane used. On this, the person blood is passed. Then it is acting like semi-permeable membrane. Then the person blood is purified here and it is collected in the bottle. Again, simultaneously it is injected into the person body with the help of vein. Such a dialyzer machine is acting like semi-permeable membrane where osmosis is taking place. And after this, the entire blood is filtered and passed into this bottle. Again, simultaneously infused into the person body with the help of vein. Already you studied in your lower standards what is artery function. Artery function pass oxygenated blood. To pass oxygenated blood, but here as it is oxygenated, but it is completely filled with impurities. What are the impurities? As we know, urea, uric acid, excess amount of vitamins, minerals, okay, then creatine, creatinine, all these are the impurities which are present in this oxygenated blood. As a result, the person is unable to respire properly. The person blood pressure become high. Okay, and the stomach bulge, complete body undergoes swelling. All these complications are seen in a person's body when both the kidneys fail, called as ESRD, end-stage renal disease. Then the person has to undergo the dialysis process. This was first discovered by William Koch, and the machine was prepared by him in the year 1947. So this machine is also working by two principles called as osmosis and filtration. Okay, the next topic, that is effects of different solution on blood cells. In depth, we will study the dialysis process in the class. Come to the next one, that is effects of different solution on blood cells. As you know, our blood cells have only all the animal cells are covered by cell membrane. All the animal cells, all the living organism cells are covered by a cell membrane, but they don't have cell wall. They don't have cell wall. That is a drawback which is there in all the animal cells, including human beings. So, as cell wall is there in plant cell, even if you keep it in different solutions, it may not show the drastic change. But here, the change is shown within a limited period of time. So, what is that is? Effects of different solution. First, it is done on blood cells. So when RBC, red blood cells, are kept in hypertonic and hypotonic solution. So first we should know what is hyper and what is hypo. Okay, hypo means low and hyper means high. Okay, so hypertonic solution is nothing but where the solute concentration is high, and hypotonic where the solute concentration is less. When such type of solutions, when RBC is kept in hypertonic solution, when RBC cells are kept for few hours, the RBC cells will undergo the shrinkage and they will die. Within few hours, we see, as you know, the lifespan of RBC is 120 days. But within hours, they will die. How they will die is when they kept in hypertonic solution, having high amount of solute in it. When the solution is prepared and when RBC cells are kept in that, the RBC cells will undergo the shrinkage and they will die. So why they will undergo the shrinkage? Here osmosis is taking place. When high concentrated solution is when RBC is kept in high concentrated solution, movement of water molecules is taking place from the RBC to the solution. As a result, they will lose excess amount of water and they will undergo the shrinkage. So such a process where they undergo the shrinkage and they will die. When they are kept in hypotonic solution, what happens? When RBC cells are kept in, red blood cells are kept in hypotonic solution, they will absorb excess amount of water from the solution outside and they will burst and they will die. Two consequences are seen when they are kept in two different solutions. In the same way, simultaneously, when you take the plant cell also, if you see the changes, you will not see such type of changes in plant cells. Why these changes do not occur in plant cell is plant cell have the extra layer which is called as cell wall. This cell wall of plant cell is made up of cellulose. This cell wall in plant cell is made up of cellulose, hence it is not showing any change. Whereas if you take any of the animal cell, here we are taking RBCs, so they will show the change. If it is hypertonic solution, highly concentrated solution, they will lose the water, they will undergo the shrinkage and they will die. When it is hypotonic solution, they will absorb the water, movement of molecules, movement of solvent from low concentration region to high concentration region across the permeable membrane, and they will undergo the bulging, they will burst and they will die. These are the two consequences we are seeing here. Okay, next live example we are taking here. The last two topics of our unit is, if you go out, you are traveling, suppose you think you are traveling in a bus, you are traveling in a car, okay, you are traveling in different vehicles, you will start feeling thirsty. As you are in a traveling position, you will start feeling thirsty. Why this happens? Why this happens? As the air is blowing on you, the excess amount of dehydration is taking place in your body. As you know, the air is liquid in condition, you will start feeling thirsty. When your thirst will start, and you will feel that I should drink cool drinks. You will feel that if I drink cool water or if I drink cool drinks, my thirst is quenched. But when you drink such type of cool drinks or cool refrigerator water and all, it will subside your thirst for shorter time. Why this happens? Especially cool drinks, you observe Maaza, Coca-Cola, Thumbs Up, Sprite and all. If you are taking for a time being, you will quench your thirst, but afterwards again you will start feeling thirsty. Why this happens means the cool drinks contain sugars mixed with carbon dioxide gas. The cool drinks consist sugars mixed with carbon dioxide gas. So when you drink it, for meanwhile only you will feel that yeah, I'm okay now. Okay, I quenched my thirst, you will feel, but after some time again you will start feeling thirsty. So avoid taking cool drinks. Take normal water, plenty of normal water. If you drink small little quantity of cool drink, you will feel that yeah, can subside my thirst. But it is not right. So try to drink plenty of water instead of drinking a small amount of cool drink. At the same time, if you go out, you will do a long journeys like temples and all. What you will feel, I should eat chips, I should eat that, I should take this, I should take. Your mother will prepare different types of food items and oily food items and taking with you to a long journey. But it is not good. Instead of taking that oily food items on like potato chips, puree and all, by seeing that attractive covers and all, children will tempt to eat. So instead of taking all those things, you better go for fruits which are having excess amount of water, hydrated fruits. Whenever you are going to the long journey, you have to take hydrated fruits with you instead of taking the oily food items like murkul, chips. That is all your mother will prepare, and sometimes you will buy also by seeing the covers and all, attractive covers and all. It is very bad habit. We should not go with that. Afterwards, it will show the consequences. Like we are suffering with emotions, vomitings, and all after coming from the journey. So avoid that. Instead of taking that all those oily things with you, you take hydrated fruits with you. What are the hydrated fruits? All majority of the fruits are hydrated. Like if you take watermelon, apple, grapes, these are hydrated fruits. You have to take with you. Instead of eating that oily food, you take that. It is good for your health and maintain the water balance in your body. Okay, so that is about our today's topic. We'll wind up with this. And before ending this topic, I want to recap the entire unit what we studied. So we studied osmosis. Osmosis we studied by taking the two activities, like we took resins, kishmish, keeping in water, keeping in sugar solution. Kishmish we are keeping in water, we are keeping in sugar solution, and we are observing the changes. If we are keeping in water, the kishmish is undergoing swelling. If we are keeping the kishmish in sugar solution, it is undergoing the shrinkage. Why this is happening? You try to find out the difference. At home, kishmish is available now. It's the best fruit to avoid corona to improve our immunity. So everybody is eating that. So what you do is keep one kishmish in water, one kishmish in sugar solution, 1 to 2 to 3, 4 pieces also you can take. Just find out the difference. You will know by that you can prove osmosis and you will remember for the longer time. Movement of molecules from low concentration region to high concentration region across the permeable membrane. What is a permeable membrane? Kishmish layer, kishmish cover. Kishmish skin is acting as a permeable membrane. Okay, that we studied first. Next, we did the potato activity. Taking the potato, making a hole in the potato, means pit groove in the potato. In that, we are adding sugar solution, keeping the complete potato in a bowl of water. Next activity, what we are doing is making same potato pit. We are making a pit in the potato, groove in the potato, and we are adding normal water, keeping the complete potato in the sugar solution in a bowl. Observe the difference. There also, where we are keeping the sugar solution, the level of sugar solution will increase in the pit of potato. That is osmosis, and another the decrease. This is osmosis. Then diffusion process. Diffusion, definition of diffusion we studied, and activities we did. Diffusion, movement of particles or molecules from high concentration region to low concentration region till the concentration become equal. Okay, we did the activity like perfume, incense stick, agarbatti, then our all outs, then room freshness. All these are nothing but these are the example, physical example of gaseous example of diffusion. Then liquid, we took one activity, that is taking a glass of water, in that adding potassium permanganate crystals or copper sulfate crystals, CuSO4 or KMnO4. Anything you can take in crystal form and add it in a glass of beaker of water. There you can see the spread of color. At a time it will not give the color. Slowly where it start reacting with the water, they will start changing the color. Uniformly it will spread the color. Such a process of movement of molecule from high color molecule from high concentration to low concentration till the concentration become equal. So in such a way we studied the diffusion activity. At home food color is available. We have seen the experiment with food color. Okay, that is about diffusion process. So how it is helping, means osmosis, how physiologically it is helping in plants. Osmosis uses in plants. They are useful for closing and opening of stomata. In plants, they are useful to absorb the water from root system to aerial part of the plant. Okay, in human beings, if you take osmosis process, that is movement of waste material in the kidneys by osmosis. Just now I explained you dialysis. Okay, then intestines, microvilli, in there also the absorption of material is taking place by osmosis process. In such a way, physiologically in all the plants and animals, that is in living organism, osmosis is helping. Next, diffusion uses. Physiologically, how diffusion is helping in the living organism's body. Like diffusion, movement of water molecule from the soil to the root system. As you know, root is absorbing the water from the soil. That process is taking place from soil to root. Absorption of water is taking place by diffusion. Okay, if you take in animal body, in our body, respiration is entirely depending on diffusion process. So when we are inhaling air, oxygen is filled in our alveoli. Our alveoli expand because of high concentration of oxygen, where in surrounding medium you will find less concentration of oxygen. So movement of oxygen is taking place from high concentration region to the blood capillaries, that is low concentration region. That is also by diffusion process, physiological process. Next, we study filtration, removing the excess amount of solute from the solution. Okay, so that is filtration process. We did the activity like I explained you. Your mother is preparing tea. Okay, and lemonade. All these activities comes under filtration process. Just with the help of stain, will remove the tea powder. So in our body also the filtration process is taking place in our kidneys by nephrons. Okay, this will you understand better in 10th class. That is filtration process we studied. Next we studied exoosmosis and endoosmosis by egg membrane activity. Egg activity. Exoosmosis means movement of molecule from inside to outside. Exo, out. Okay, exoosmosis. Endoosmosis means movement of molecule from outside to inside. That is endoosmosis process. Exoosmosis and endoosmosis process we study. Okay, then next, hypertonic and hypotonic solution. What is the dialysis? We study Then we studied the reverse osmosis and what is Graham's law of diffusion. We studied in this topic. Then what are the important questions expected from this is all the activities are very important, right? The activities in your class work, activity of osmosis, 2 activities are there. How to write the activities? Already I said you the format. Aim you have to write. Apparatus required you have to write. Precautions you have to write. If chemicals are there, write the chemicals separately. Then procedure you have to write. Then observation you have to write. At last you have to write the conclusion. So write all the activities in your classwork. Expected questions are write the activity to prove osmosis. Any activity you can take. Which activity are potato activity. Sometimes they give in bracket write potato experiment means you have to do potato experiment. The next write the activity to prove diffusion. Then write the activity to prove filtration. Then write the activity to prove exo and endo osmosis. Write the activity to prove exo and endo osmosis. These are the 4 mark questions. Then the last 4 mark question is explain the functions of plasma membrane or write the functions of plasma membrane. These are the 5 questions, 4 mark questions we are expecting from this lesson. All the activities. Okay, diffusion activity, osmosis activity, filtration activity, exo and endo osmosis activity. Then functions of plasma membrane. These are the 5 important questions we are expecting from this lesson. Then what are the 2 mark question is write the differences between diffusion and osmosis. Write the differences between diffusion and osmosis. Okay, then write the functions of osmosis. Write the functions of osmosis, 2 marks. Write the functions of diffusion, 2 marks. Then what are the 1 mark important question we are expecting from this topic is define diffusion, define osmosis, what is dialysis, who prepared the machine for dialysis. Don't think that it is an lecture part, they will not ask you. Then what is reverse osmosis, where reverse osmosis is taking place. What is reverse osmosis? Reverse osmometer. These are the expected 1 mark question from this topic. So, if you want, write it in the question answer form or directly, you can write as I said you, keeping the concept in mind. Okay, so with this, we find out our topic, and if you have any doubt, you are welcome. Yes, if you have any doubts, please keep it in your chat box.